Good morning, everyone. My name is Mariam. Today I'm going to talk about improving fertilizer nitrogen use efficiency using concentrated urea bands and with genotype preference for nitrogen forms. I'd like to acknowledge my co-authors and my honors supervisors who are shown in underline, as well as the institutions who supported this research. And finally, I would like to acknowledge the Soil Science Society for their generous support, which has allowed me to attend this conference. In developing the hypothesis of this research, I examined ways to minimize nitrogen loss pathways. In broadacre agriculture, fertilizer nitrogen is applied as urea. The urea undergoes hydrolysis to ammonia, and this process raises the photosphere pH, impacting on the equilibrium between ammonium and ammonia. As the pH increases above 8.2, more ammonia accumulates, and if this process occurs close to or on the soil surface, volatilization losses are likely. In the experimentation that I undertook, urea was banded 75 millimeters below the soil surface, so volatilization could be minimized based on the research by Rushard and others 2014. Typically, ammonium is quickly transformed to nitrite and to nitrate, and what I was interested in achieving was to store the mineral nitrogen, a greater proportion of the mineral nitrogen in the ammonium form. I achieved this using concentrated urea bands, which store nitrification and store greater proportion of the mineral nitrogen in the ammonium form for longer. This strategy of using concentrated urea band was based on the research by Passiora and Wetzeler 1972. In the diagram shown on the left, the delayed nitrification effect is achieved as the central part of the photosphere becomes an unfavorable environment for soil microbes involved in the nitrification process. And this effect is attributed to a high osmotic gradient, high pH, and high ammonium and ammonia in the photosphere. If DMPP is added to urea, shown diagrammatically here on your right, this potentially extends the period of nitrification inhibition and may result in even higher ammonium to nitrate ratios. Returning to nitrogen loss pathways, we can see that nitrate, which is negatively charged, is vulnerable to leaching, and in coarse soils, it can be leached beyond the root zone or into deeper depths where crop nitrogen recovery is poor. Another nitrogen loss pathway occurs in oxygen-limited conditions, often resulting from waterlogging. In this environment, soil microbes would use oxygen molecules from nitrite and nitrate, causing denitrification. In the research I undertook, I examined methods of stalling nitrification so the greater proportion of mineral nitrogen could be stored in the ammonium form. And the reasoning behind this approach was to minimize potential losses via volatilization, leaching, and denitrification. In my experiment, I also used two wheat genotypes, Yitby and Frame, and they were selected based on their differential responses to ammonium or nitrate. The genotypes I just spoke of were selected based on research by O'Sullivan and others, 2016. They measured shoot dry weight, which is shown here on the y-axis, and supplied either nitrite or ammonium to Yitby and Frame, shown here on the x-axis. When Yitby was supplied ammonium or nitrate, it grew more biomass in response to ammonium, and conversely, Frame produced more biomass when in response to nitrate. I thought that these points are important considerations when examining nitrogen use efficiency. My hypothesis, therefore, was fertilizer nitrogen use efficiency can be improved by the use of concentrated urea bands and with genotype preference for either ammonium or nitrate. And the methods I used were subsurface banding of urea with and without nitrification inhibitors, and the wheat genotypes I indicated were selected based on their contrasting performance when supplied with ammonium or nitrate. The treatments selected were subsurface urea banding, included urea only as the industry standard, and urea plus DMPP to delay nitrification, 2 wheat genotypes, frame and Eastby, and I chose a range of different nitrogen rates to develop response functions, and I anticipated that the concentrated urea band effect, which I referred to previously, would occur at higher nitrogen rates, 90 and 120 kilograms of nitrogen per hectare. The measurements were undertaken on samples collected 52 days after nitrogen banding and sowing. That included shoot dry weight, shoot nitrogen fertilizer uptake, and soil residual ammonium and nitrate. I also determined the critical external nitrogen requirement to achieve 95% of maximum shoot dry weight. And for the purpose of this study, nitrogen use efficiency was defined as the nitrogen required, critical external nitrogen required to produce 95% of maximum shoot dry weight. This is an important point, so I'm going to elaborate it for the moment. Here on this graph, you can see shoot dry weight on the y-axis and nitrogen rates on the x-axis. The mean of the replicate data are shown as the black squares, and the response function is fitted as dashed line. When we take 95% of maximum shoot dry weight, where it intersects the response function and is projected to x-axis, we can identify the nitrogen rates required to produce 95% of maximum shoot dry weight. Moving on to results. Here we have shoot dry weight on the y-axis and nitrogen rate on the x-axis, and we are examining the response of wheat genotype frame, which produced more biomass when supplying nitrate in O'Sullivan's research. If we have a look at frame shoot dry weight in response to banded urea plus DMPP, we find that the nitrogen rate required to produce 95% of maximum shoot dry weight was 60 kilograms of nitrogen per hectare. However, for the banded urea treatment, it required 112 kilograms of nitrogen to achieve 95% of maximum shoot dry weight. If you look at the same graph for Yitby, it shows Yitby required 86 kilograms of nitrogen per hectare to produce 95% of maximum shoot dry weight when urea plus DMPP was banded, and only 62 kilograms of nitrogen per hectare when urea was banded. To summarize these results, it will be useful to look at them in a slightly different context. The table results here show that there is no significant difference between the maximum shoot dry weight in any of the nitrogen banding treatments. However, what's different is that the nitrogen required to achieve 95% of maximum shoot dry weight was significantly less when urea plus DMPP was banded. And this is explained by the lower amount of fertilizer nitrogen uptake required to achieve that biomass. There are no significant difference between residual nitrates 52 days post-harvest. However, the residual ammonia was significantly higher for the higher nitrogen rate. When we consider Yitby, There was also no significant difference in shoot dry weight in response to banded urea treatments. However, the nitrogen required to produce 95% of maximum shoot dry weight was significantly less when urea was banded. And this again was explained by the lower fertilizer uptake required to produce 95% of maximum shoot dry weight. As for frame, there was no significant difference between residual nitrate on the E2 post-harvest. However, once again, the residual ammonia was significantly higher for the higher nitrogen rate. So my key findings were the most efficient treatment required approximately 60 kilograms of nitrogen per hectare to achieve maximum shoot dry weight, 95% of maximum shoot dry weight, and were achieved by frame where urea plus DMPP was banded and YIT-B where urea only was banded. These nitrogen use efficiency results were partly a function of the amount of fertilizer nitrogen uptake required to produce 95% of maximum shoot dry weight. To conclude, I would say that the critical external nitrogen efficiency is a function of the interaction between banded urea treatments and which is likely influence nitrate to ammonium ratios, and the efficiency of wheat genotypes for specific forms of mineral nitrogen, which means that research that examines nitrogen use efficiency needs to consider that the results obtained may be genotype specific and could also change with the predominant form of the mineral nitrogen supplied. These factors impact on internal use efficiency of mineral nitrogen and nitrogen loss pathways in soil. That concluded my talk. I would like to acknowledge Charles Sturt University, New South Wales Department of Primary Industry, and the E.H. Graham Centre. I'd also like to acknowledge Dr. Tiffy, Mrs. Sharon Nielsen, and Dr. Lockett for statistical support. And just as a note, on Thursday afternoon, Chelsea Jenke will be presenting on a complementary area of research that I just presented. Any questions? Any questions? Yes. Yes.
Thanks. Thanks for your presentation. I wondered, first of all, the residual and ammonium and nitrate, is that residual in the soil?
Yes, yes.
Okay. Why the ammonium was higher in the treatment without DMPP?
Yeah, so the DMPP primarily added to the urea that we use to, as I said, hold nitrification process, which means that we would expect that we have a greater proportion of the mineral nitrogen in the ammonium form. Yes. So we would expect that result, higher ammonium. And also, for the higher rates, we would definitely have higher residual ammonium.
Sorry, I thought in the table it was higher in without DMPP.
I can't find it. So, yes, for the...
okay, that one was high. Okay. But in the framework, ammonia was higher without DMPP.
Yes.
And so, Mariam, I think the question is it's rate specific. So if you look wherever the higher ammonium is, there's a higher rate being applied to achieve 95%. So there's a rate effect coming through.
My name is Jian. I'm from Natural University. Thanks to all of you who are coming to my talk. And today, I would like to share some of my recent findings on how the soil organic amendments affect the transpiration efficiency of wheat plants in acidic duplex soil. So before I look into the transpiration efficiency, and I would like to start my talk with a very important plant structure known as stomata. These are tiny openings or pores on the surface of leaves, which allows for gas exchange. So it allows the plant to take in CO2, and it's also open gate for water loose through transpiration. This also explains why there is a close link between the plant biomass production or grain yield and the transpiration. High biomass production is always associated with high transpiration because the stomata conductance or openings tend to increase to maximize the CO2 intake. Unavoidably, it will cause more water loss. So under water limit conditions, there is a need for a tighter trade-off between the CO2 intake and water loss. So this is where the definition of the water use efficiency has been introduced. It reflects the plant's capacity to convert water into biomass. It is defined as the amount of biomass produced per unit water use, which is the slope of this line, and is called water use efficiency or transpiration efficiency. These two terms have been used interchangeably by people, but they are different. They merit at different levels or different scales. At the field level, it is called water use efficiency. It's the ratio of total biomass to evapotranspiration. The water loss here is a combination of evaporation from the soil surface and the transpiration from plant canopy. But when the water use efficiency is measured at plant level or in the pot level, it is termed transpiration efficiency. It is a ratio of biomass to water transpired. So the water loss due to evaporation has been stopped or minimized by covering the soil surface with a thick layer of plastic beads. When the transpiration efficiency is measured at the leaf level, it is termed instantaneous or intrinsic transpiration efficiency. It is a ratio of instantaneous CO2 assimilation to transpiration rate. In this study, we will focus on the transpiration efficiency at the plant and the leaf level. Unlike water use efficiency, which can be increased by many management practices such as residue retention or fertilization, which can maximize the proportion of water transpired relative to water loss due to evaporation. But the transpiration efficiency at the plant or leaf level is relatively a constant or static term for given crop species in a given environment. This is why more than 70 years research showing limited evidence of any direct effect soil management on transpiration efficiency. And there are some unpublished studies, unexpected results from our lab, have consistently shown that transpiration efficiency can be increased by deep application of pollinator into the sodic clay subsoil. This leads to the aim of this study. We want to confirm these findings, and we also want to provide a possible explanation by focusing on the stomata conductance and the function, and see how the function of stomata in response to the addition of different organic amendments. They have 5 amendment treatments, including control with no amendments, and we also have inorganic fertilizer control. This will be compared with other 3 organic amendment treatments. The straw will be mixed with inorganic fertilizer in liquid form at the same rates as the NPK's treatment. And because the nitrogen release from the pollinator is too slow to meet the plant requirement in the later stage. So this is why we have additional treatment where the pollinator has been mixed with urea. So we want to make sure that the nutrient, especially nitrogen, is not going to limit any positive effect of organic amendments, such as biological or physical amelioration effect. This photo was taken at the tillering stage, and you can see all these 3 organic amendments that produce the more tiller number more tillers, and this resulted in a relatively higher shoot-draw rate than the inorganic fertilizer control. Of course, the poultry, they had produced more tillers, but because they suffer from severe nitrogen deficiency after stem ligation, so that's why at harvest and bolting stage in the biomass is similar to the NPK's treatment. But the final transpiration efficiency was consistently higher for all these three organic-based treatments than this inorganic fertilizer control. These results actually has been repeated for at least 3 experiments. But in this experiment, we also made the transpiration efficiency and the leaf level using the LCI portable photosynthesis system. And we did the measurements for every 2 days and for maybe at least more than 30 days. And early stage, under well-watered conditions, or in the later stage during the dry and rewetting cycle. And this instrument can provide detailed information on the gas exchange, which includes the CO2 assimilation rates, the stomata conductance, and the transpiration rates. And then we can calculate the instantaneous transpiration efficiency based on the CO2 assimilation and the transpiration rates. And for these 3 selected key treatments, and you can see this organic amendments can increase the CO2 assimilation, and which can explain why these organic amendments can produce more tiller numbers or produce higher biomass at early stage. But thus the instantaneous transpiration efficiency, they show a slight increase, but they are not significantly different. When the plant is under moderate water stress conditions, by withholding the water for more than 48 hours, the stomata conductance decreased from 0.8 to 0.1. And this can explain the significant decrease in the CO2 assimilation and the transpiration rate. But this transpiration efficiency nearly doubled that of under well-watered conditions. This is in line with the previous findings that plant roots are able to sense soil dryness in response by synthesizing hormones like abscisic acid to induce stomata chloride and to decrease the transpiration rate while increase the transpiration efficiency. So this happened for all these 3 amendment treatments. But when you compare among these treatments, the organic-based amendments actually induced more reduction in stomata conductance, and so they should lower transpiration rates and higher transpiration efficiency. And this is under moderate water stress conditions. But even after watering, few hours after watering, you can still see there's a difference among these treatments. And the stomata conductance has recovered quickly for this inorganic fertilizer control, but it should delay the recovery for this organic-based treatments. And so this explains the relatively lower transpiration and higher transpiration efficiency. Apparently, this difference among treatments should not be attributed to the difference in soil moisture content because this is a few hours after watering. The soil moisture content is very similar. So what other soil conditions can explain this difference in stomata functioning? How about the soil biological effect? How about its biological effect? We did extract microbial DNA and we did the sequencing for bacteria, and this figure shows the relative balance of bacteria and genus level. And you can see Bacillus, which is a well-known genus, consists of many strains of plant beneficial bacteria. They are dominant in this soil. They were slightly higher in this organic-based amendments than inorganic fertilizer treatment. But we cannot say they are more abundant in this treatment because it's a relative balance. It really depends on the balance of other genes. So this increase here may simply due to the decrease in other genes. So this is why we also calculate the absolute bacillus balance by multiplying the total copy number of bacteria with the corresponding relative balance. And now we can say that this bacillus is more abundant in this nutrient-rich organic minimum treatment. And also, we have another control with no plant and no amendments. So when you compare it with this, with and without plant treatment, you can see the bacillus was more abundant in the presence of plants. This would indicate the important role of the roots or root extracts in attracting this plant beneficial bacteria, such as bacillus. This has been confirmed by a positive correlation between the root length density and absolute bacillus balance. And we also found a positive correlation between the transpiration efficiency at plant level and absolute bacillus balance. How are we going to explain this positive correlation? Our study consistently showing that additional organic amendments can increase the transpiration efficiency in this sodic duplex clay subsoil. And the organic amendments can also increase the balance of bacillus, a plant beneficial bacteria. And this increase in bacillus is actually linked with the organic amendment-induced increase in root proliferation. And it may also partly attributed to the organic amendment-induced improvement in soil physical structure, especially the improved soil aeration may facilitate the colonization of the roots by this plant beneficial bacteria. And then the increase in the bacillus can trigger the synthesis of hormones such as plant growth promoting substances, which can explain the vigorous early shoot and root growth and increase in plant biomass and increase in transpiration efficiency. On the other hand, there are many evidence suggest that the inoculation of plant roots with bacillus can result in increase in leaf hormones under water stressed conditions. And this can result in the more reduction in stomata conductance and decrease in transpiration and then increase in transpiration efficiency. So this is how the increase in the bacillus balance of plant beneficial bacteria linked with the increase in transpiration efficiency and plant level. Of course, the process highlighted in the red is based on the literature. We don't have the direct evidence from this study to support. So the further study is still required to verify this complex link between the organic amendments, plant beneficial bacteria, leaf hormones, and transpiration efficiency. Finally, I want to thank to our funding body, GRDC, and thanks everyone who has contributed to this study, and thanks you for your attention. Thank you.
Hello everyone, my name's Hayden Jones from Waikato Regional Council in New Zealand. I've been asked to chair this session. We do have a couple of questions come through for our speaker on the app. The first is, would sulphate of ammonia be a better choice when using poultry litter?
Sorry, Jenna. I will leave my video off because I have poor connection here. So John, can you repeat your question again?
The question was, would sulphate of ammonia be a better choice when using poultry litter?
Because normally, to my knowledge, the poultry litter is the nitrogen form is dominated as ammonium. There is not like a compost which is dominated as nitrate. So we did measure the ammonium concentration in the poultry litter. It is very high.
Thank you. And just one final question. What would be the N response, NUE, by the two cultivars when only nitrate N was applied?
Sorry. So what's the cultivar? Are you asking about the cultivar? The wheat cultivar?
By the two cultivars.
I only have one cultivar, so which is EP, and this is I only have one wheat crop species.
Okay, all right, thank you. We better move on to the next speaker. Next speaker is Frederick van der Boom from University of Queensland.
Yes, all right. Thank you very much for your attention. I'm indeed a postdoc at the university. Thank you very much for your attention. Yeah, feedback's gone? It's gone. All right. Well, thanks again. I'm a postdoc at the University of Queensland. I've arrived in Australia about 2.5 years ago. Originally, I'm from the Netherlands, where, you know, I guess everybody knows that our yields are fairly high. Our systems are very high intensity. And so when I moved here, I had a bit of a shock, sort of realizing how complex soil systems are. And so it's a real joy to be in Australia and work with what are so many super interesting interactions and dynamics that I don't really see at home. So the setting that I work in is one that is driven by let's say breeders who are developing crops with deep roots. And the idea is that if you have a crop with a very deep root system, you might be able to tap into deep water reserves that you wouldn't access otherwise. So the idea is if you're in a system such as the dryland systems here in much of Australia, if you're in a situation with terminal drought, you might have yield benefits if a plant can still access these deep resources. Now, the issue with the, let's say, breeding for these crops is one that strikes me as a little bit peculiar, which is that much of the selection actually goes by proxy traits. So it's extremely difficult for breeders to actually do selection of these genotypes in the field. And so what happens generally is that, for instance, plants are grown for 5-7 days in a pot. There's a selection for root angle, for instance, which is narrower or wider. And then that is supposed to deliver crops in real life that have deeper root system. Now there are some correlations that have been found in greenhouse systems and all that, that suggest that this might be true, but I'm wondering whether that actually happens in real life in the field. And particularly, the systems I work with on the Darling Downs, I work in vertisols, they're highly complex, they're way more complicated than just issue of access to water. So I imagine that having deep roots would work very well in a system where everything else is available and the plant has nothing to fear from the perspective of nutrients or so. But the reality is much more complicated. So the systems I work with, we have declining soil fertility, so the subsoils are very much starting to get depleted. Farmers are forced to apply phosphorus nowadays. They're typically banded, which is related to moisture dynamics. Topsoils are getting more stratified. We have no-till systems, which have been going on for 20-25 years. So what you basically get is you have this continuous depletion of the subsoil and an enrichment of the topsoil. But at the same time, it can get rather hot out there and the topsoil dries out pretty fast. So plants have a short early season period where they can access that phosphorus, in this case, in the topsoil, which is immobile. They might have a little bit longer for getting phosphorus out of bands, but altogether, it's not as straightforward, I think, to develop crops with deep roots and to just ignore those details of the system. And so I have a lot of questions, basically revolving around: are deep roots actually going to work in real life conditions. Yeah. So that basically, here's a small summary of a whole bunch of questions. I actually have a lot more, and it's probably a lifetime quest to solve some of these. But essentially, if you have deep roots, those deep roots, is this crop going to miss out on the stratified nutrients? What about plasticity? Here on the left side is an example. It's old work from the 70s, but we know that roots have specific responses when they encounter phosphorus in the soil. So if that's a response, is that going to override roots going deep? What about wet years and dry years? Is the benefit of deep roots going to work in years where it's very wet? Etc., etc. On the other hand, you might have the opposite of deep root system. If you can select for a deep root system, you can also select for a shallow root system. And so the shallow root system might actually be better at getting that phosphorus out of the topsoil. It might have benefits of early access. We might have feedback there, because if it takes up phosphorus very early, maybe the root system will be larger by virtue of early feedbacks. Yeah. So I have a lot of questions, and this is what I've been working on for the last 2.5 years, roughly. But as I said, it's probably a lifetime thing. Yeah. So what I've been doing the last 2.5 years is I'm trying various approaches, including rhizoboxes. I use an automated lysimeter system that you can see on the right side. I go out in the field, I do field samplings. We just planted some small variety plots in the field. And essentially what I do is I collected two contrasting genotypes from breeders, one with a narrow root angle and one with a wide root angle, and they're supposed to deliver deep or shallow roots. Yeah. And so then I play around with phosphorus placement. I play around with a drying topsoil, which is interesting because of this drying and early season dynamic. And I play around with some starter pea. So I'm hoping to give you a little bit of an impression of what I do exactly. And I'm hoping to tease you a little bit to think about all these issues that are actually real life problems. And so I think we sort of need to get out of the lab sometimes and see how stuff works in the field. So here's an example of some responses I get in my rhizoboxes where I put phosphorus as starter or as a deep band. And I suppose you can guess roughly where I might have put the phosphorus exactly. This is just to illustrate that root systems are very dynamic, and they can respond to let's say, the heterogeneous system that is soil. So it's important to understand not only sort of in theory the predefined architecture, but to understand actually how that interacts with real life situations. Yeah. Here's another example of what I get in my lysimeters. So these are two durum genotypes that I've grown until flowering in this case, with those same treatments where you have a dry control, a dry band, wet topsoil. And this is just an example of what kind of differences I get. And this is very much phosphorus response and not so much a genotypic response. So you get this huge difference from the dry control, which is very much depleted in phosphorus, all the way to the right side, where you have phosphorus that I mixed all the way through the profile, high fertilization, and this is the difference you get. So the right side is rather artificial because obviously the plant has everything in great supply. The reality is that in the field we're probably somewhere between a dry band and a wet topsoil. So there's a lot of potential to get more out of our systems, and the question is whether these genotypes, these root systems, can deliver some of it. Here's another example seen from the top in my lysimeter system. So just as an illustration, I really get huge differences, and this is all phosphorus driven. Yeah. All right, so some actual data. This is on the left side my lysimeter trial up to flowering. Those are what the photos were from as well. And so I have my basic control treatment on the left side. And then the next one is where I banded phosphorus at about 25 centimeters. The topsoil, where I put it in the top 10 centimeters dispersed, so a fertile topsoil, but nothing in below. And mixed treatment, where phosphorus is dispersed all the way through the soil profile. And then the sort of bluish, purplish ones are the ones where water was available all season long. And red, yellow ones are the ones that dried out. And there's a lot to unpack here, and probably a lot more than I can actually discuss right now, so I'm happy to discuss afterwards. But in general, what you see is that there's a very clear phosphorus response that, in my mind, is much larger than the actual genotypic differences. Although we do get genotypic differences, particularly in the topsoil, I think is quite striking, where if you look at the blue bar, the two blue bars, the right one is the wide genotype and the left one is the narrow genotype. We see that the wide one, that is a sort of a topsoil forager, is much better at grabbing that phosphorus that is only located in that zone compared to the narrow one, which goes deep fast. Generally, this is also what we see in the riser boxes, which are a little bit shorter season, although I find the interesting effect there with where phosphorus is banded, where a narrow genotype seems to be able to grab phosphorus and that band slightly earlier. So I see an early effect of the way the soil is explored, but that largely disappears on the longer periods. So that I'm not showing it here, but I can tell you that there is a phenotypic delay that is caused with the interactions with phosphorus, and so the benefit that I see of a narrow genotype initially quickly diminishes or disappears. This is a figure of the early root responses, and there's a lot to unpack here, and I probably, you probably shouldn't display this kind of complex figures in the presentation, but I'm doing it anyway. Quickly, I want to point out two things. First of all is the control treatments, which are marked WC and NC. On the bottom left, you can basically identify them quite easily. And so this is the distribution of roots down the profile from 0 to 60 centimeters. And what you basically see is that the narrow and the wide genotypes both don't have a lot of roots down there when there's not a whole lot of phosphorus around. As soon as you add phosphorus in whatever form, the amount of roots at depth sort of increase. So it's very vital to have phosphorus available to the crop, even to express that genotype in the first place. The other part is what I would like to show is the band zone specifically, which coincides with this early exploration thing. So if you look at the band zone on the left side, you basically have the two controls, narrow and wide. And the yellow ones that are the wide treatment, you can see that two boxes are very close to each other. And this is the wide control and the wide band. Yeah. So there's basically no difference between the wide control and the wide band. On the opposite, you have the narrow control, which is the most left one. And there's a rather large response there with the band, which coincides with this proliferation that I showed. So there's a lot going on, and there are a lot of interactions. That's basically the biggest message that I wanted to get across. Now, I was inspired by yesterday's presentations about rock, scissors, paper, so there you go. I'm not going to ask you to do rock, scissors, paper with your next, but generally my observations are that there is a sort of a rock, paper, scissors thing going on here. So when you have root architecture that might work, or the root architecture might be different in soils, but if there's not enough phosphorus nutrition, then that architecture doesn't really express. So phosphorus nutrition beats architecture. Yeah. In a similar way, architecture might actually help for drought, which is the theory that is behind all of it. I haven't really been able to confirm this in my type of experiments, but the theory holds pretty well, I think. And then lastly, drought beats phosphorus nutrition, particularly when phosphorus is located in the topsoil. So if the topsoil dries out quickly, you can have the most beautiful fertile soil, but if there's no water, you can't access it. So we have very clear rocks, paper, scissors going on. So to conclude, there's a lot to be done, and there's way more than I can do alone. So I'm asking you to help and to think about these kind of issues. Yeah, we need to understand very well in what situation genotypes work and how management and the environment influence that. Yeah? So even in the Northern Grains region, we have a massive amount of climate variability. And some parts, some things might work and some things might not. And we need to understand that in real life, we need to get out of the lab, get out of gel systems and all that. And we need to understand how this stuff works. And then we need more methods. Washing roots is a difficult task, and so we need new ideas to get this stuff done. And so I'm hoping that one of you might come up with a good idea at some point. Thank you very much. I didn't mention this. In this case, we took photos of the root system, and I've analyzed them using WinRISO, where you get length out of it, but also area. I found that I get good correlation between them, but at the higher end, length tends to top off, which is related to overlap between the roots. So I prefer to present intensity, which is essentially the visible amount of area. But it's very closely correlated with root length. It's root area, visible root area, so it's surface. Yeah.
We don't really have time for questions. Sorry, you can move on.
Thank you very much.
Thank you.
Thanks very much for the opportunity. My name is Bing Han, and from the University of Melbourne. My work is very much about converting lignite into fertilizer, and today I'm going to talk about a portion of my work, which is the lignite ammonia adsorption and surface chemistry after dewatering. So what is the problem we want to address? So ammonia emission is a serious environmental pollution, can cause water eutrophication, odor and soil acidification. And ammonia can also be oxidized in the atmosphere to generate nitrous oxide, which is a potent greenhouse gas. And the alkaline ammonia can react with acidic pollutant, such as sulfur dioxide, to generate particulate matter, which can lead to the serious human disease. And the ammonia emission is also a huge waste of resource because the manufacturing of the ammonia is energy intensive. 5% of the world's natural gas production and 2% of the world's energy was consumed just by the synthesis of ammonia. So what's the mitigation strategies? So we have to look at the life cycle of the nitrogen in the animal manure because the animal feedlot is a hotspot for the ammonia emission. Basically, the urea in animal manure hydrolyzes into ammonia, and the ammonia can be lost into atmosphere through volatilization. And it can also be oxidized into nitrate and nitrate and lost as, through leaching. And in previous research, people basically apply different inhibitors, such as the urease inhibitor and nitrification inhibitor, to block the pathways of the hydrolysis and the nitrification. Here, we would like to use lignite as an adsorbent material to retain the nutrient in the animal waste. So why do we choose lignite? Lignite is a low-rank coal which has a very low energy value. It has a high moisture content, low ash, and it also has a high humic acid and a high CEC. So those properties make it an excellent material for ammonia adsorption. And the lignite is abundant in Victoria, for example, in the Latrobe Valley accounts for 25% of the world's lignite reserve. And on top of that, the lignite also have many benefits. For example, it's non-hazardous and it can be used as a soil conditioner, which increases the crop yield. However, there are still some challenges for the lignite. For example, the high water content of lignite increases the transportation cost, so it's not economically viable. And the self-combustion of lignite is also a serious issue because it's related to the safety of storage and transportation. So first, we would like to investigate how to dewater lignite. The simplest way to dewater lignite is just applying heat. So firstly, we conducted a thermogravimetric analysis. Basically, there are 3 stages. Below 100 degrees C is the dewatering stage. It's basically the water content rapidly decrease. And between 100 and 300 degrees C, it's a stable stage because the organic matter doesn't change much. However, if the temperature is above 300 degrees C, it's a volatilization stage. The organic matter reduced significantly mainly because of the decomposition. Because the organic matter is a very valuable nutrient, so we think that the temperature range between 100 and 300 degrees C is a reasonable temperature range for dewatering. So we basically applied different temperatures between 100 and 300 degrees C to the lignite. And so you can see the first figure shows that after air thermal treatment at 200 degrees C, the uptake of aqueous phase ammonium increased by 65.8%. And the second figure shows that the gas phase ammonia absorption in moist air on treated lignite increased by 28.8%. And the third figure shows that the gas phase ammonia absorption in dry atmosphere on treated lignite increased by 311%. So basically, this result tells us the dewatering of lignite actually can enhance the ammonia absorption, and 200 degrees C is the optimal temperature. So after we got the performance data, we would like to look at the mechanism underlying the performance. We firstly did some surface chemistry characterization. Basically, the FTIR analysis reveals that the carbon-hydrogen bond was transformed into a carbon-oxygen bond during the lignite dewatering. And the XPS data indicates that the carboxylic ratio increased from 6.2% to 7.5%. So this means that this suggests that the oxygen-containing functional group actually play a very important role in the ammonia absorption process. So to gain more details about the mechanism, we use Boehm titration to quantify the oxygen-containing functional groups. And there are basically 3 types of functional groups, and all of them increase first and then decrease with increasing temperature. And 200 degrees C is the optimal temperature. And the pH value of the lignite material also shows the same trend. So this positive correlation between the absorption performance and the number of oxygen-containing functional groups confirms the functional group play a very important role. And however, a common sense in the absorption research area is that the surface area play a very important role. I would like to show here, this is not the case in this study. Basically, the helium-ion macroscopy indicates the pore structure was destroyed after the dewatering process, and the nitrogen absorption reveals the surface area actually decreased after the dewatering. So this means that it is the functional group rather than the surface area that facilitates ammonia absorption. So we already know that oxygen-containing functional groups play a very important role, but we are trying to figure out which functional group is the adsorption site, because there are at least 3 types of oxygen-containing functional group in lignite. For example, hydroxyl functional group, the carboxyl functional group, and lactonic functional groups. we found that the linear correlation between adsorbed ammonium and total concentration of acidic functional groups on the lignite was very significant, whereas other correlations for single groups were poor. So this means basically all the functional groups have a contribution for the ammonia adsorption, and also the slope of linear regression for total concentration of oxygen groups was close to 1 on a molar basis, suggesting that one oxygen group interacts primarily with only one ammonium ion. So basically the adsorption side was identified. So on top of that, reducing the risk of spontaneous combustion is also very important because it's directly related to the safety of the transportation and the storage of lignite material. Here we did some analysis. The result is that after thermal dewatering at 200 degrees C, the parent activation energy of the combustion increased from 34.8 kilojoule per mole to 37.8 kilojoule per mole. This means that dewatering can actually reduce the risk of spontaneous combustion for lignite. So this will improve the safety of transportation of the lignite. So we have demonstrated the dewatering process can be successfully used to enhance the ammonia adsorption. We wonder whether this technology can be extended to the other coal material because lignite is mainly located in Victoria region, but the black coal material is available all over the world. For example, Queensland has a lot of black coal mines. So yes, the result is very positive. After the modification, we use similar air thermal treatment. The ammonia gas uptake by black coal increased by 11-fold. Is almost as efficient as the lignite. So for future work, currently we are using low-quality thermal energy to dewater lignite, but in the future we would like to use solar energy because solar energy is free. Actually, Australia enjoys some of the highest value of solar irradiation in the world. So our hypothesis is that we can convert solar irradiation into reactive oxygen species, and those species could generate oxygen functional groups on the surface of lignite, which can increase the ammonia uptake. So that is my presentation, and thanks my supervisor, Professor Deli Chen, Professor Yizheng He, and my main collaborator is Dr. Clayton Bartley and Ms. Fei Zhang, and here are some references in case you are interested in the research. Thank you very much.
Thank you, Bing. So our next two speakers are pre-recorded. Next up we have Casey Dillette talking about mapping nutrient availability in large intact soil cores, and followed by Wendy Quayle talking about optimising the management of poultry litter in Australian cotton production.
So most soil nutrient analysis techniques are destructive and rely on chemical extraction. So I'm going to talk about a GRDC project that I'm working on that uses non-destructive techniques to visualise the distribution of potentially available nutrients in the soil, in particular in large soil columns. So first I'll just give an overview of the overall GRBC project, then specifically talk about UniSA's part in the project, which is to develop large diffusive gradients in thin film devices, or DGTs, for mapping potentially available nutrients in large soil cores. So I'll present some results that we've collected from synchrotron analysis of these DGTs, and also how we've optimised these DGTs at such a large size, and further optimisation that we're undertaking. So overall, the project aims to contribute to Australian rainfall profitability by using synchrotron approaches. And it's a partnership between University of South Australia and the University of Queensland, and of course, where we carry out most of our analyses. So ultimately, we hope to collect soil cores from the field and examine the root distribution in these impact soil cores, and then compare this to changes in nutrient availability and also changes in the distribution of organic matter and the form of organic matter. This information is really crucial for understanding how to close the yield gap in constrained soils, that be acidic soil or nutrient depleted soil. And all of these analyses that we carry out are done at a large scale, and that's really the advantage of synchrotron analysis compared to lab-based techniques, where most X-ray analysis techniques, which is what I'm focused on, are done at a much smaller scale. So really the advantage of synchrotron analyses are that we can have much larger imaging. So whether that be tomography, looking at the root architecture, which you can see here in the middle slide, in the middle picture in the next slide, or large images of DGT devices. So the focus, as I mentioned, here at UniSA is developing techniques for large imaging of DGT devices. So there are several researchers and several beamlines involved in this project, each with a particular focus. So Dr. Helen Howe does most of her research at INBL, which is a medical beamline, and she's developing pipelines to look at root distribution and how it's affected by fertiliser placement. Dr. Han Wang does most of his research at the infrared beamline, and he's really focused on how changes in the forms of organic matter impact soil fertility. And most of my research is done at the X-ray fluorescence beamline, where I image DGT devices with the ultimate goal to improve fertiliser placement and application strategies. So my work is closely connected with Helen. So Helen carries out root tomography to look at root architecture. I then section the soil cores and then examine the nutrient distribution and see how these two things overlap. So firstly, what is a DGT? So a DGT is a way for non-destructively measuring the available nutrient pool in the soil. The DGT measurement incorporates the initial soil solution concentration of that particular nutrient of interest, as well as the ability of that soil to resupply that nutrient in response to its removal. But really, the principal theory behind the DGT is that it connects the plant roots. So the DGT acts as a nutrient sink for the nutrient or analyte of interest. Typically, first I'll just from. So this is the general structure of the DGT device. So it consists of a plastic casing, and then from top to bottom, this is the part that's in contact with the soil. So it consists of a filter membrane, a diffusive layer through which the analyte of interest diffuses, and a binding resin which binds the nutrient of interest, which you can see here. So the blue layer is placed in contact with the soil. And this is the general structure of a DGT device. But what you notice is that they're generally typically quite small DGT devices, around 2 centimetres squared. So in order to look at the distribution of nutrients throughout the soil core, we needed to develop something much larger, and that's been focus of my work. And usually for DGTs, they're typically by eluting the binding layer and then calculating the mass of the analyte that has accumulated in that binding layer, what we're doing is looking at the spatial distribution of nutrients in the resin rather than evolution. And we're using synchrotron-based XFM to do that. So why use XFM for DGT analysis rather than some other imaging techniques? So another common way would be to use laser ablation, which is shown on here. The laser ablation RCPMS has several advantages, is that it can be done at quite high resolution, down to about 4 microns. It's quite easy to calibrate, there's minimal sample preparation. But the main disadvantage is that it's really quite slow. So to map anything larger than 1 centimetre square takes a number of hours. Another option would be to use benchtop X-ray fluorescence microscopy or benchtop XRF. However, this has a much lower sensitivity compared to synchrotron-based XFM. So overall, the main advantages to synchrotron analysis are that it's much faster and that a much lower resolution and much more sensitive compared to other available techniques. So as I mentioned before, commercial soil DGT is relatively small with a surface area of only about 1 to 2.5 centimetres square. So our project initially last year was focused on making quite large DGTs and working out what materials are best to make them from and sort of stand up best to the XFM analysis. So we tested a range of DGT gels and different binding agents to look at their different properties. So that picture there just shows the preparation of the large DGTs and their deployment in soil information studies. So there were 3 parameters that we considered. So the handling properties of the gels, any shrinkage and drying effects, and analytical constraints. So this table shows how most of them fare. So we looked at 3 gel matrices. So we looked at acrylamide and a polyacrylamide gel. So polyacrylamide is probably the most commonly used gel type in DGTs. And we also used a polyurethane gel and a number of different binding agents which bind either cations or anions. So Helix binds cations, so most of the micronutrients such as zinc, which is what we investigated, whereas ferrihydrite and titanium dioxide are anion binders. So we were looking at the binding of phosphate. And as you can see, we then investigated handling properties, shrinkage and drying properties, and analytical constraints. And what we found that the polyurethane gels, the 2 polyurethane gels, were superior to the other gels. And so we have then used these in subsequent experiments. So just presenting some results from our experiments with these large polyurethane DGTs. We conducted a small incubation study using a range of different granular fertilisers in a clay cropping soil, neutral pH of around 7.3. And after incubating these fertiliser granules for 28 days, we then applied DGTs of 10 centimetres by 17 centimetres on this soil to look at how the nutrients had diffused from the fertiliser granules, if indeed they had diffused. And just to point out again that DGTs only measure the potential available pool of a nutrient. This is some results from that experiment. So you can see on the left, that's a picture of 3 different DGT devices mounted on a frame, the XFM being one. From the left, there's a polyurethane gel. In the middle is a very high acrylamide gel, which you can see by the yellow colour. And finally, a polyacrylamide gel, which you can see the drying properties are not plastic for that because there's quite a number of holes in that gel. So we discounted that gel for subsequent experiments. The best gel we found was this polyurethane gel. And on the right is an XF image of that gel that shows the distribution of zinc, the distribution of potentially available zinc as a result of deploying that DGT on the granules that were incubated in the soil, as shown in the previous slide. And you can see potentially available zinc has diffused around 2 centimetres from that site of application. So another part to this work was, well, one of the limitations is that although synchrotron analysis is much faster than benchtop excipients analysis, with so many gels which need to be analysed because so many field soils have different properties, we need to increase the throughput of analysis of these gels. So we actually developed a way to analyse these gels in the background of experiments. This is just an image of the beamline, and there are two detectors on this beamline. So what we did was analyse the DGT gels upstream. We analysed the DGT gels upstream here, and then the beam comes down through here, showing how the white line. And then more samples can be analysed down here. And we tested a range of gels and found that it doesn't affect, most of the gels don't affect beam properties. So this can be used to increase the throughput of the XFM beamline because DGTs can be analysed upstream without any penalty to experiments that happen downstream. Once we had found the best DGT, we then used these large DGTs to look at the distribution of available nutrients on our soil pores. In this case, it was repacked soil pores, where we used two soil types, a vertisol, a carousel, and again, granular fertilisers, an MAP, and a co-granulated zinc MAP fertiliser as well. And I'll just show you the initial data that we've got from this experiment. So I should point out, before we did this work, Helen at INBL examined the root architecture of the wheat plants that were grown in these wheat cores, and then I vertically sectioned these cores and deployed DGTs, which you can see here, to look at the distribution of macronutrients and micronutrients. And this is the DGT deployed on the soil core. So in the far right picture, that shows the distribution of potentially available zinc. You can see this bright spot in the middle, and that corresponds to higher concentrations of zinc in the DGT, and actually corresponds to near a location where one of the near the fertiliser granule band was placed. So unfortunately, you can see that for phosphorus, we're not able to get a signal. So we think this comes back to analytical constraint. So unfortunately, the fluorescence emission for phosphorus has quite a low energy, and quite a lot of that signal is absorbed by air, but we think quite a lot is being absorbed by the DGT material itself, so the polyurethane itself. So our next experiments are looking at improving this binding lacquer to improve the phosphorus signal, so we can also map the distribution of macronutrients as well as the cationic nutrients, such as zinc. In summary, polyurethane-based gels are most suitable for large DGTs, and synchrotron XFM of these layers is possible to map the distribution of potentially valuable micronutrients. And by carrying out analysis in tandem at the XFM beamline, we can increase the throughput of that beamline, meaning that we can analyse many DGTs in samples without penalty to that beamline. And the next step is deployment of these DGTs on intact field cores, which is happening in emission. Thank you to our collaborators and funders, in particularly GRDC and also Australia. Thank you.
Hello, my name is Wendy Coyle. I work for Deakin University in the Centre for Regional and Rural Futures in Griffith in New South Wales. The title of my talk today is Optimising the Management of Poultry Litter in Southern Cotton Production. I'd just like to take a chance to acknowledge the collaborators and partners on the project. The Cotton Research and Development Corporation has provided funding for which I'm sincerely grateful. The National Landcare Programme Smart Farms Initiative has enabled me to be able to collaborate at a national level with Dr. David Rowlands, Queensland University of Technology and others to develop this work into a farmer tool app which can be used to manage organic amendments on farm and which is currently being developed through a project called Unlocking the Value of Soil Organic Amendments. I'd also like to make special thanks to local collaborators, the Irrigation Research and Extension Committee, which is a grassroots grower organisation in the Murrumbidgee irrigation area, Cotton Info, the extension arm of the cotton industry, and then also the individual farmers around Griffith and Darlington Point, which are so generous with their time and their knowledge and all the farm operations. All of this work is being carried out at commercial scale on farm. And finally, I'd also like to thank the staff within my group in Griffith, particularly Dr. Jackie Webb and Dr. Rakesh Awali. So we're talking about cotton and poultry production in the southern Murray-Darling Basin region in the Murrumbidgee and Lachlan valleys, where approximately 30,000 hectares of cotton is now being produced, up from about 5,000 hectares in 2013. And the reason for this increase is because of the good cotton price, but also because of genetic developments in cotton, which has enabled it to be grown in sort of non-traditional, cooler southern areas as intensively as further north. Simultaneously, there's been a rapid increase in poultry production to meet domestic consumption. So this graph shows increases. In 1966, there were 20 million chickens slaughtered in New South Wales, and currently there's now been more than 200 million chickens being slaughtered. This was reflected around Griffith with a big jump in production around about 2016, and the volumes of chicken litter bedding produced went from 4,000 metres cubed per week to more than 10,000 metres cubed per week every week. And at regular application rates of about 10 metres cubed per hectare, which is what farmers tend to use, this amounted to 52,000 hectares of land that could be amended. And our rough estimates indicate that with this amount of chicken litter, it amounts to about 3,000 tonnes of available nitrogen every year. So how can we use these nutrient resources within chicken litter, which is in close proximity to the farmers and which therefore make it potentially viable to use within their crop nutrient budgets? For them to have predictability, we need manure composition, soil nutrient availability, crop growth characteristics, target yields and crop requirement, environmental conditions, and probably most challenging is to get a handle on mineralization rates or the sinks for nutrient release from amended litter. So a little bit about mineralization. We can use a theoretical basis to estimate nutrient release from amended litter through lab incubations, and there's been a lot of work done on this. But problem is these lab incubations are done under very controlled moisture and temperature, which don't actually represent field conditions. And so the factors that we use from lab incubations don't necessarily follow. So we can use field trials to get field conditions and use availability by grain yield differences in experimental setups, and also plant nutrient uptake differences between fertilized and controlled plots. The problem with doing this on commercial farms is that, and at scale, is that the crops need to be responsive to nutrients of interest, which is often quite difficult when you've got crops in rotational sequences and management decisions being made year on year as seasonal conditions change or as budgets change. We can also use probes and cation exchange resins, which are put into the soil over the season and take up plant available nutrients. And we've been doing some of this work with plant root simulator probes provided by Western Ag in Canada, isotope labelled urea, which is the work of Dr. Jackie Webb. But I'm not going to talk about that today, but I am going to talk about a field trial which we've had running at the Witten Field Station, which is about 30 kilometres south of Griffith. It's at commercial scale, with all the operations being carried out by farmers. And the hypothesis is that there's a 1 to 3 year response to the rate of manure application for nutrient uptake, nutrient use efficiency, and cotton yield. The litter or manure can offset the need for pre-plant fertilizer with respect to yield and cotton fibre quality, and that manure can provide residual effects for soil chemistry and microbiology that are identifiable by soil testing, and therefore can be economically recognisable for cotton nutrition, in order for farmers to be able to decide whether applying manure is worth it and at what rates are the best rates to use. So the field trial consists of a randomised control block design with 3 replicates and 6 treatments. The treatments consist of a farmer practice conventional urea, mineral-only nitrogen treatment, a reduced mineral nitrogen treatment, and then 4 tonnes, 8 tonnes and 16 tonnes per hectare of chicken litter in combination with urea to achieve matched target N rates. The methods for the poultry litter were the on-farm manure piles were sampled 10 times, and then samples were taken from each part of the heap, and then they were combined to provide composition. This is the composition of the poultry litter that we use for each litter each year. The values do change, but generally the litter around Griffith at the moment is the 2019 values, with nitrogen values of 4.1 to 5%, and high values of ammonium, substantial amounts of phosphorus, and important for cotton is high amounts of zinc. The 2017 sample was a little bit different in that it was more composted, indicated by higher values of nitrate, and the other nutrients in a little bit more concentrated form. So the soil type at the Witten Field Station is a transitional red-brown earth, indicated and classified by Van Dyke. It's locally known as a Mundaware clay loam. It has a light loamy texture in the upper part of the profile in the A horizon, down to about 18-20 centimetres, although clay is starting to extend up due to cultivation. This data is from September 2015, before the trial started, and it followed barley, and you can see that there's not very much N in the profile. It's been drawn down by the cereal. There's high amounts of Caldwell P in the upper part of the profile. Increases typically to high pH with depth, and acidity isn't a problem in the surface, but it's starting to come in at depth as well. Results for cotton yield showed that when we compared integrated chicken litter and urea applications with conventional urea at matched N rates, there was no difference in yield between chicken litter amended and non-amended plots in the first year of the trial. Yield increased at the highest rate of chicken litter at 16 tonnes per hectare by 1 bale per hectare, which was significant in the second year of the trial. By the third year of the trial, lower litter amendment rates had also increased, but the highest rate had not increased any further. So this suggests that a new equilibrium may have been reached, and that any further increases in yield, which could potentially occur through potential yield of these varieties, was being restrained by underlying soil fertility conditions, physical conditions, or seasonal conditions, or irrigation. By the end of 3 years, at the highest rate of chicken litter amendment, the farmer would have been $2,000 per hectare better off. Cotton lint quality is also an important parameter for cotton farmers, because they get paid depending on the quality of the cotton being within certain ranges. There was no difference in cotton quality between treatments, but there was differences between years, which is indicating that it's caused by seasonal differences or irrigation. However, the bottom chart does show that there's some almost significance according to treatment in micronair, which is one of the important quality parameters. And although all of these are well within the base range and there would have been no price penalties incurred, I think this data does show that by manipulating nutrition using manure and mineral fertilizer in these southern regions, differences in quality can be achieved. And this could be important if quality issues do become important in a particular season, if the season isn't that great. These graphs show the changes in potassium and phosphorus in post-harvest 0 to 30 centimetre soil samples for each particular year, and they are conservative for the application of litter in that they have good relationships between litter rate and soil concentrations, and therefore seem to be fairly reliable soil tests to ascertain the value of litter in the longer term for farmers. Phosphorus is looking like, particularly Caldwell P, is looking like it is accumulating in soils, and so by taking soil tests, this might have to be managed appropriately. The residual patterns for mineral N being accumulated or carried over with respect to different rates of chicken litter aren't as clear, and this is probably because we've got different rates of chicken litter in combination with urea and making assumptions on nutrient release rates. We've got plants taking the nitrogen up as the main driver of nitrogen that might be left in the soil, and we've also got environmental losses. pH, cation exchange capacity, and zinc also didn't really change over the period of the trial according to the treatments. For the soil biology, we did see increases in microbial activity as measured by standard methods according to litter rate, but we didn't see the same consistent changes in biomass carbon. And by the end of the trial in 2020, there was no significant difference in biomass carbon in the highest rates of litter compared to mineral fertilizer or the control, although the mineral fertilizer plots did show lower amounts compared to control and amended treatments, which suggests it may not have been as conducive an environment for microbial communities. Just to put some economics around it, when you look at this table, it compares the value of having to pay for nutrients in litter as compared with nutrients in mineral fertilizer, and you can see that depends on spreading and freight costs, but at the current amount you're looking at probably about $100 cheaper to deliver the same amount of nutrients from chicken litter in this region compared with mineral fertilizer. So in conclusion, manure composition is variable, but for consistent production from local poultry facilities, the farmers who are in close proximity to sheds of particular processing may be able to get some uniformity of product. Around Griffith now, the sheds are producing about 80 kilograms of available nitrogen and about 26 kilograms of available P for every 10 meters cubed. At practical application rates of 50 meters cubed per hectare, these can therefore substitute for pre-plant urea N and replace mineral P for a 10 to 12 bale cotton crop. On transitional brown earth riverina soils, yield can be increased by about a bale per hectare in the second to third year of application after cereals. After 3 years of application, residual soil effects are evident in phosphorus and potassium, and these tests may be used to be able to determine the residual effects of, maybe it's used as a surrogate for the residual effects of litter, to put some economics around it for the farmers. But there is an indication that K and P are starting to build up, and this might need to be managed. The benefits for soil biology were unclear, and similarly carbon, although we only looked at TOC, there was no changes. Using chicken litter at 50 meters cubed potentially can save about $300 per hectare in mineral fertilizer and increase your return by about $2,000 per hectare in 3 years. Thank you for your attention. I hope you enjoy the rest of the conference, and yeah, have a good day. Thanks.